In this video, we're going to learn about osmosis. So, when you start your elementary science, like from your SS1, SS1, yeah. So you do a topic called the osmosis and diffusion. This is very important. Okay, so I've made a video on diffusion already. So you can go to my channel and watch the video on diffusion. So today we're going to learn about the definition, types, and examples on the osmosis. Okay, it's very important for one to understand some basic principles. You just imagine that if you soak a beans in water for some times, you discover that the beans become swollen. Okay, so yeah, this a green a beans that are used to prepare nice meal and stuff like that. So most of the time you can soak it. There's some African made food when you make, if you want to make something like I think moi moi. Yeah, you tend to soak the bean for some time. Most of the time, you just soak the bean if you want to make moi moi to remove the coats surrounding the bean. But in the real sense, if you soak bean, what happens? The bean tends to swollen. So what actually make it to get swollen when you put it into water? All right. So the answer is what osmosis. So an osmosis is essential for all living organism. Okay. So once the water gets into the beans, what happens? This bean become turgid. When you say turgid, it means it fills with water and becomes swollen. That means what osmosis of actually what occurred. Okay, so a clear definition of osmosis. So it's very important to get your paper and pen or your book and pen and write this information down. So osmosis is the movement of water. When the water through, very important, selectively permeable membrane is very important. When something is membranous, it means it allows some substances to pass through it. Okay, so from a dilute, when something is dilute, means it's not highly concentrated. Okay, from a dilute solution to a more concentrated solution. All right. So for this membrane, it would be nice for us to understand what membrane is, given different types of what membrane. Okay. So here I'm going to check the first types of membrane. So we'll have what we call permeable membrane. So a permeable membrane is a membrane that allow all substances, both the solutes, both the solvents, to pass through it freely. Okay. So a very good example of a permeable membrane, you have your filter paper. I always forget the name of this. This is a filter paper. It's permeable. It allows substance to pass through it. Then you have your cotton wool. Cotton wool is permeable. Yeah. Once you put water through cotton wool, what happens? It pass through it easily because you have a lot of pore spaces that can allow substance to pass through it. Then just imagine, just assume a permeable membrane, just like a gate. A gate that's open that allows anybody to pass through it easily. Okay, just assume, just an analogy for you to understand these concepts. Okay, so the other type of membrane you have here, you have your semi-permeable, or you call it partially permeable membrane. So which allow only certain substances, especially water molecules, to pass through it. Why they block what other larger substances from passing through it. Okay, so for example, like your cell membrane, okay, your cell membrane allows some substances and prevents other larger substances from passing through it. Okay, just like for example, analogy, like a gate to pass the gate that allow only personnel, only authorized personnel to pass through it, but other or unauthorized people will have no access to pass through it. So that's just an analogy to understand. So how about something impermeable? When it's impermeable, it means it does not allow any substance to pass through it. So, for example, like a brick wall is impermeable because nothing can pass through this brick wall. Okay, so that is that. So we have one other type called selectively permeable membrane, just like we define in osmosis. So this is a membrane that allows water molecules to pass through it, but restricts some dissolved substances. Okay, some dissolved. And a good example of this membrane, you have a cell membrane, you have your dialysis tubing, you have your egg membrane. So it prevents some dissolved substances. Okay, just like for example, like this your sieve. So if you put you can put substances that have a lot of spaces. It allows some of them, but prevents some to pass through it. Okay, so just kind of an analogy. All right. So there's some terms that is very important to understand in osmosis. So water molecule. So water molecule is very important because you cannot discuss about almost osmosis without making reference to water molecule. So water molecule, these are particles that move during osmosis. So this is the water molecule. Then you have another term called the solutes. So a solute can be a substance that is mixed with solvents. For example, a solute can be a salt. Okay, so a substance that dissolves in water, such as a salt or a sugar. These are known as a solute. So these are solutes. Okay, so when you put them into a solvent, what happens? It dissolves into a solvent, and the solvent mostly in science, in universal solvent is water. Okay, then what we call the solvent. So this is a liquid that dissolves the solute. Okay, it's a liquid that dissolves the solute, which is what water. All right, so it's a very good example of what a solvent. Okay, so condition necessary for osmosis. What are those conditions that makes osmosis to occur? All right, so osmosis can occur when water molecules are present. So water molecule has to be present for osmosis to occur, and a selectively permeable membrane separates two solutions. So it's important, and there's the difference in water concentration between two sites. So it's very, very important for all these things to be present before osmosis can actually what occur. All right. So now I want us to understand different types of solution. So we'll talk about water as a universal solvent. So we have different kind of solutions that affect osmosis. I'm going to make a video on experiment on osmosis, but I'm not going to make it in this video. Okay, where we're going to learn about how different solutions affect osmosis. All right. So now the first type of solution here will have what we call the hypotonic. They will have the hypotonic solution. So in this type of solution, we have what they call the low solute concentration is very low. Okay, it's not highly concentrated, and you have high water concentration. There's high amount of water and little amount of solute concentration. And water tends to enter into the cell. And when water is getting into the cell, what happens to the cell? The cell tends to become swollen. Just for example, you are drinking a lot of water, drinking water. You're satisfied with keep drinking water. How happen? You get extremely filled up. That's what this thing is all about. So the cell become what swollen. So you can see this picture here. So in this hypothetical solution here, you can see, for example, if a lot of water get into the red blood cells, what happen? It tends to become what swollen. And if it keep getting swollen, what happen? It can burst. Okay, even in the vacuole. So the vacuole can become highly turgid. When it become turgid, it means it's filled with what is water. Okay, so for example, we have your distilled water, which is a good example of a hypotonic solution. So the next type we have what we call the hypertonic. So in the hypertonic solution, we have higher solute concentration, higher amount of solutes, for example, like a salt, which is present, high amount of salt is present. Then you have what water concentration. So you have more salt and little amount of water. So it makes that solution very, very what harsh. And what happens? Water tends to what to leave the cell. So we have high amount of solutes, you have low amount of water. What happens to the cell? Water tends to leave the cell, and this causes about what shrinking. So the cell tends to what to shrink. When some of the storage is very something is swollen, turgid, then once it shrink, what happens? It becomes what flattened. Okay, just like for example, if you carry a balloon, you blow air into it, it becomes what turgid because it's swollen. But if you remove the air out of it, what happens? It becomes what flattened. So we call it a shrinkage. Okay, so you can see this cell become what shrinked because why water molecules are what left the cells, and look at the vacuole becomes smaller because why water molecules are what left the cell. Okay, so this solution is a very hard solution, and a good example of this is what is solution that have high amount of salt content. Okay, or high amount of what sugar present. These are an example of what hypertonic solution, which is strong solution that affects cells. So the next one, the third one is isotonic. So isotonic is just kind of a moderate amount of both high solution and in the low solution. So you have equal solution of solute concentration on both of them, and you also have no net movement of water. Okay, and the cell remain the same size, no changes, nothing happens. All right, a good example is just 0.9% of sodium chloride. So this means the amount of salt in water is what is a minimal amount. So this the solution is what is moderate, and There's no form of the cell being bursting because of turgidity, or the cell become flattened because water tends to leave the cell. So it just what normal. Okay, so now we're going to go through osmosis in plant cells. Okay, so now we'll see how osmosis impacts plant cells. So in a hypotonic solution, which is what a is not a strong solution, a solution that has to do with distilled water. Okay, what happens to the plant cell is water tends to enter the cell. Okay, and the vacuole, when it enters the cell, the vacuole tends to expand, and this is a vacuole here. This is an example of a plant vacuole. What happens to the cell? The cell become turgid. So once water enters the cell, what happens to the cells? It become what turgid. So it's got swollen like this. And what happens when you get turgid? It help the plant to keep upright. Okay, the plant tends to become upright and they function properly. That's the effect of hypertonic solution in plant cell. Okay, but in a hypertonic solution, which is a strong solution, water tends to leave the cell. Okay, and the cytoplasm shrinks from the cell. It shrinks, and what happens? The plant tends to lose a lot of water, and if it's occur for a long time, what happens to the plant? The plant start wilting. They wilt, and the plant may eventually die. And this process is called plasmolysis. Okay, so you can see this picture here. This is a normal cell. Now this is a turgid cell that have lots of water. You can see it's swollen. Then this is a plant. This is a plant cell that shrink because why water have left the cell. So if this occur for a very long time, this plant will tend to lose. The plants will tend to become wilt because why water have leave the cell, and the plant may eventually will die because of lack of water. So we have isotonic. So in this, there's no net movement of water, like I told you, just moderate, and the cell just becomes flaccid. Okay. All right. So osmosis in animal. How does this affect animal? All right. So for this, you can see that in a hypotonic solution, the same thing occurs just as the plant. Water enters the cell becomes swollen, and the cell may burst depending on the amount of water. So it can burst if the content of water is very very high. Okay, so this process is called osmotic lysis. All right, so you can see this picture in hypotonic solution. So water tends to get into the cell, and the cell become what turgid. Okay, so in an hypertonic solution, which is very strong solution, this water tends to leave. All right, it tends to leave the cell, and the cell shrinks, and this is called what crenation. All right. So what happens to the cell when the cell has a high amount of such solution? What happens? It tends to shrink, and this is called what crenation. So this is very important terms you need to understand it. But one thing about biology is that biology has a lot of terms. So just for you to get familiar with just terms. So in isotonic solution, what happens to the cell? The cell just maintains its shape because everything is minimal. Okay, very important. Okay, now let's see the importance of osmosis. So why does osmosis actually matters? Why? One, it helps plant to absorb water. Plant should to absorb water because plant tend to absorb water from the soil for them to perform well. And this helps the plant to maintain its cell shape, and it keeps the plant firm and upright. So once it's upright, it can easily trap sunlight for photosynthesis. And it regulates water balance in living organisms, just like the term you use is hemostasis. Just like in osmoregulation, regulating the water balance in an organism. Okay. Then why osmosis matters? So it aids the movement of water between cells. So water is very important. We can't do without water, and this helps kidney function by absorbing water. The kidney needs water. All right, and it also assists in seed germination. You can't put your seed on the soil without watering it. If you leave it there, nothing's going to happen. But once you apply water to it, what happens? It tends to germinate. So it's very important. So just the difference between osmosis and diffusion. So in osmosis, only water molecules move, but in diffusion, any particle can move. So the next one, it requires a selectively permeable membrane, but in diffusion, a membrane is not required, is not needed. Osmosis only occurs in liquid, but diffusion occurs in liquid and in gas. And for osmosis, from higher water concentration to lower water concentration, while from diffusion is from high concentration to low concentration. So that's the difference between osmosis and diffusion. Okay, so let's just round everything up with just some exam multiple choice questions. Okay, so osmosis is the movement of what? It's not sugar molecules, it's not oxygen molecules, it's what? Water molecules. Okay, very important. Then osmosis requires a thick cell wall, no sunlight, chlorophyll. It requires what? A selectively permeable membrane. Then when a plant cell is placed in distilled water, what happens to the cell? When you add distilled water, it tends to become solid, yeah, swollen and turgid. So it become plasmolized. No, become flaccid. No, become dead. No, it becomes what? Turgid. Okay. So I'm going to end the video here. So just keep watching my video. And if you watch the video to this point, thank you very much. And just keep on my channel because my next video I'm going to explain about plasmolysis. I'll explain plasmolysis. I'll explain hemolysis, and after which then I'll just give a clay experimental setup of osmosis, how it's been done. All right. So see you next time, and thank you for watching my video to this point.